Hello everyone, in this video tutorial we will talk about the protein and their metabolism. As we know that our body is mainly made up of protein, and to maintain a normal health it is estimated that around 1 gram of protein per 1 kg of a person's body weight should be consumed. So protein are not stored for later use, so excess protein must be converted into the glucose or triglycerides and used to supply energy or build energy reserves. Now there are five major elements present in the elemental composition of proteins. The number one is carbon, which is 50 to 55%, hydrogen is 6 to 7.3%, oxygen is 19 to 24%, nitrogen is 13 to 19%, and the last one is the sulfur, which is 0 to 4%. As we know that the proteins are made up of amino acids, and there are 20 different amino acids present in our body. Amino acid, now what is amino acid? Amino acid are a group of organic compound containing two functional groups, amino and carboxyl. The amino group is basic while a carboxyl group is acidic in nature. Each amino acid has a central carbon atom which is known as alpha carbon. There are four different groups attached to the alpha carbon. These are amino, carboxyl group, and the hydrogen, and the last one is the R group. R group are variant in nature. If we change the R group, then it means that the amino acids are changed. This R group, which is variable, it is either hydrogen or alkyl group. Due to this variable, the amino acids are different from one another. Now we comes to the protein structure. So the first protein structure was determined by the Sanger in 1951 in insulin molecule. So the first one is the primary structure. In a primary structures, many peptide chains are arranged in a linear sequence. Okay, the second one is the secondary structure. In a secondary structures, when a polypeptide chains of amino acids become spirally coiled, so this structure is the secondary structure of a protein. The third one is the tertiary structure. When the secondary structure of a protein is arranged into a three-dimensional structure. So here is a three-dimensional structure which is known as tertiary structures. And when two or more polypeptide chains are arranged into a large size, then it is called quaternary structures. Okay, in the quaternary structures we have a very famous example, that is hemoglobin, which are quaternary structure protein. Okay, now what are the functions of protein? Protein perform the most important functions in the life of living organisms. The first one is the structural. So the collagens and keratins are the protein which provide structural components. The second function is the contractile. So the myosin and actin or the proteins which help in the movement of muscles. The third one is a transport. So protein role as a transport is the hemoglobin and lipoprotein, which are proteins, and they carry essential substances throughout the body. The protein role as a storage is the two protein which is known as cysteine and ferritin, which store nutrient. Proteins are hormones like insulin, growth hormone, and we know that the hormone regulate body metabolisms and nervous system. And enzymes are also protein in nature, which catalyze biochemical reactions in the body or in the cells. So secretin, trypsins, they all are enzymes which are protein in nature, and proteins recognize and destroy foreign substances, so they have a role in protections. And fibrinogen which are proteins and they recognize and destroy foreign substances, so there are many functions of a protein. Digestion of proteins begins in the stomach. When the food are passed through the esophagus and enters into the stomach, so here pepsin and HCl break down the food into the small particles. So the chyme are formed in the stomach and this chyme when enters into the small intestine, so here bicarbonate and digestive enzymes are present in the small intestine. The bicarbonate neutralize the acidic HCl, okay? And the digestive enzyme aids in the digestive process. Pancreas also release some enzymes like trypsinogens, chymotrypsinogens, okay? And there is an enzymes in the wall of small intestine which is known as enterokinase, which activates trypsin, which in turn activate the chymotrypsinogen. These enzymes liberate the amino acids. And then these amino acids transport across the intestinal wall in the cell. And there is a transporters which is sodium amino acid transporter, which transport the amino acids in the bloodstream, okay? And the nitrogen waste that is liberated in this process are converted into the urea in the urea cycle. Now, what are essential amino acids and non-essential amino acids? So, essential amino acid cannot be made by the body. They must come from the food. There are 9 essential amino acids: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. These are essential amino acids. They are 9 in number. And there are 11 non-essential amino acids which are produced by our body. If we do not get it from the food we eat, so they are known as non-essential amino acids, which are 11 in number. They are alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. Okay. Now, what is amino acid pool? Amino acids from different sources, like dietary protein, like body protein. So, from different sources, when mixed up, they constitute an amino acid pool. So from different sources, when amino acid mixed up, so they constitute what is known as general amino acid pool. So every adult body contain 100 grams of free amino acids, which is known as amino acid pool. So this pool has no anatomical reality, means that there is no specificity of an amino acid to make a protein. So there is no anatomical reality here, but only represent an availability of amino acid building unit. So when protein mixed up, so they constitute what is known as general amino acid pool. Now, there is a term which is known as protein turnover. The replacement of older protein is known as protein turnover, as they are broken down within the cell and the new protein are formed. So the whole phenomena is known as protein turnover. Different types of protein have very different turnover rates depending on their particular function. Like structural protein have a half-life period, and enzymatic protein have a shorter lifespan. So different proteins have a different turnover rates, okay? Here some proteins which are degraded fast if it contains sequence of proline, glutamine, serine, and threonine. So these proteins are degraded fast. Now there are some proteins in this table and they have a different turnover rates. Like collagen, they have 117 years, islands have a 70 years, replication factor C subunit 1, they have 9 hours. 40S ribosomal protein S8, they have 3 hours. Ornithine decarboxylate have 11 minutes. So different types of proteins have a very different turnover rates. Now, what is transmination and deamination in the protein metabolism? So that what is transmination? So transmination is the transfer of amino group from amino acids to a keto acids. What is keto acid? Amino acid without amine group is known as keto acids. So this is known as transmination. So first of all, we have an amino acids, and this comes from dietary protein, body protein. So this amino acids converts into the keto acids. What is keto acid? Amino acid without an amine group. Okay, so this is known as transmination, in which amino acids are converted into the keto acid by an enzyme which is known as aminotransferases. Okay, so this keto acid have a different role like energy generation, synthesis of fat, synthesis of glucose, synthesis of non-essential amino acids. Okay, so what is transmination? In which the transfer of amino acids into the keto acid. All amino acids except lysine, threonine, proline and hydroxyproline participate in the transmination. Now, what is deamination? Deamination is the removal of amine group as ammonia NH3. Okay, so in the process of transmination, the amino group of most amino acids are transferred to alpha ketoglutarate. Okay, so so this alpha glutarate to produce glutamate, and this glutamate serves as a collection center for amino group in the biological system. So glutamate is the only amino acids that undergoes divination to liberate free NH3 for urea synthesis. Okay, so this is the whole sketch of transmination and deaminations. So here from dietary protein and body protein, amino acids are formed which converts into the keto acid by an enzyme aminotransferases. Okay, so these keto acids have a different role like energy generation, synthesis of fat, synthesis of glucose and synthesis of non-essential amino acid. Now, what is deamination? Deamination, when in the process of transmination, the amino group of most amino acids are transferred to the alpha ketoglutarate. Okay, so they form glutamate, and this glutamate serve as a collection center for amino group. Okay. So glutamate undergo deamination to liberate free NH3 for urea synthesis. Okay. Now, in the last, what is urea cycle? The urea cycle is a cycle of biochemical reaction that convert highly toxic ammonia to urea for excretion. As we know that glutamate is the only amino acid that undergo to liberate free ammonia, so this highly toxic ammonia converts to urea for excretion. So this whole cycle is known as urea cycle. About 10 to 20 gram of ammonia is removed from the body of a healthy adult every day. At this functional urea cycle would mean excess amount of ammonia in the body, which can lead to hyperammonemia and related disease. It means that when there is an excess amount of ammonia in our body, it can lead to hyperammonemia and many other diseases. Site the urea cycle take place in the liver, and the first two reactions occurs in the mitochondria, and the next three reactions occurs in the cytosol. This is the whole structure of the urea cycle. 5 different enzymes are used in the cycle. So the NH3 will react with the CO2. Here an enzymes carbamyl phosphate synthase 1 are used, which are activated by the N-acetyl glutamate. 2 ATP are consumed here. So they form carbamyl phosphate, and this carbamyl phosphate react with ornithine to form citrulline, and inorganic phosphate are released, and here enzyme ornithine transcarbamylase are used. This product citrulline is transported to the cytosol. Okay. Now citrulline, when react with aspartate, they will form arginosuccinate by an enzyme arginosuccinate synthase. Okay. So and here also 2 ATP are consumed. From arginosuccinate, there are two ways. The one is arginine and the other is fumarate. And here enzyme arginosuccinate lyase are used. So from arginine when react with ornithine, there is a urea formation occur by an enzyme which is known as arginase. From fumarate, there is a malate. There is a conversion to they convert to malate, then to oxaloacetate, which are translated to regenerate aspartate, which are required for reaction 3. Okay. From fumarate, which convert to malate, there is a generation of 1 LADH. Okay, which is equal to 2.5 ATP. So this is the whole cycle where 5 different enzymes are used. In the last, there are diseases which are related to the urea cycle imbalance. So what is hyperammonemia? Hyperammonemia occurs when there is deficiency of more of the urea cycle enzymes. So hyperammonemia 1 is the deficiency of carbamyl phosphate synthase 1. Okay. So the second disease is the hyperammonemia 2, which is the deficiency of ornithine transcarbamylase. The third one is the cytolinemia, deficiency of arginosuccinate synthase. Okay. The fourth disease is the arginosuccinate aciduria, which is the deficiency of arginosuccinate lyase. And the last disease is the argylemia, which is the deficiency in the argillase enzyme. So this is the whole about the protein metabolism.